Hello! So today I want to talk about one of my favorite topics, and that's endosymbiosis. I think this topic is really cool, and it's really interesting to think about in terms of evolution. So, the endosymbiosis theory is the theory that at least mitochondria and chloroplasts are actually descendant of bacteria that were engulfed by eukaryotic cells, and then through a process, we'll talk about a little bit whether or not it was parasitic or mutual, became mitochondria and chloroplasts, or at least mitochondria, it might have been parasitic. There is a paper that I want you to read certain parts of that talks about whether or not that origin of the mitochondria was parasitic at first or if it was mutual. Right now, right, as of like present time, it is a mutual relationship, but it may have started out parasitic. So that's the whole idea behind endosymbiosis. And above me this way, you can see the endosymbiosis theory written out in this form. What I want to do with you is I want to actually go through how this process formed and essentially the different layers and membranes that are involved in this understanding this theory and sort of how that theory came about. All right. So I have a blank slide next, and I want to draw it out with you. Okay. So if you're going to draw this with me, I need you to have a couple of separate colors. Right. And the first thing I'm going to do is I want to draw a little organism, right? This is eventually going to become our mitochondria, right? But this is a bacteria, okay? So we have our bacteria, right? And then I'm going to use blue to draw out a eukaryote, okay? So we're going to say this is the nucleus, right? And this is our eukaryote, okay? And so what happens, at least in the first part of this, this phase, is this eukaryote engulfs, using phagocytosis, this bacterium, okay? So it's going to engulf it, all right? So this is our eukaryote, right? And it's going to engulf that bacterium, right? And then for whatever reason, that bacterium doesn't disintegrate. It's not eaten by that eukaryote and instead is living inside of it. And again, this could have been parasitic, right? Or it could be mutual. This bacterium, right, creates energy, right, using oxygen, okay? So this eventually turns into, right, this eventually turns into a eukaryotic cell, right, that now has a nucleus, right, has a nucleus, and inside of it there is a organelle, right, that now has two membranes around it. That first membrane, the outer one, so the inner membrane here is the bacteria membrane, right? That's the membrane that was around the original bacteria, okay? That next membrane that is outside of it, but still part of that organelle, right? This is the phagocytosis membrane, right, of primary endosymbiosis. And I say primary endosymbiosis because you guessed it, there's going to be a secondary endosymbiosis later, but not with the mitochondria. Okay. So this organelle now has two membranes around it, right? And eventually this, right, becomes the mitochondria. Right? We can draw this again in a different form, and instead of that bacteria being an aerobic bacteria that does cellular respiration using oxygen, that could also be a bacterium that does photosynthesis, right? Does photosynthesis, right? Most likely it was a cyanobacteria. Okay? And so in that case, this would be, right? This would be a green bacterium, right? And then we would have our green chloroplast, right? With our second layer, right? And our nucleus, right? So now this would be a chloroplast. Oh, you can't see that. Let's go this way. Right? All the layers would be the same, so that bacterial layer would still be the bacteria that came from the original cyanobacteria, and then you have your phagocytosis membrane surrounding that that organelle. Okay. So if you have questions about that, you can let me know. We're gonna go to the next slide and talk about the evidence for this. Okay, so some of the supporting evidence is that mitochondria and chloroplasts are about the same size as a bacterium, right? They both replicate via fission, as do bacteria, right? So they both don't undergo mitosis like eukaryotic cells do. Instead, they replicate inside of the cell, and they do that via fission, right? They have the double membranes, consistent with that engulfing mechanism via phagocytosis that we just talked about, right? They do still have similar genes inside of them that bacteria had, and they're circular, right? So they're in that circular format instead of linear like a eukaryotic gene would be, right? And those genes code for their own replication, right? And when we sequence those genes, they actually turn out to be more similar to bacterial genes than to eukaryotic genes. And the paper that you're going to read talks a little bit about mitochondria and the genes that they've sequenced from mitochondria and how that relates to current bacteria that are living and what that informs about how those mitochondria might have evolved. Right? So this is the paper that I need you to read, okay? So originally, right, and currently, right, we know that mitochondria are mutualistic, right? We know that mitochondria and the cells that have mitochondria in them have a mutualistic relationship where the cell itself will provide oxygen and the mitochondria will use that oxygen to create ATP that then functions to power the entire cell, right? So that is a mutualistic symbiotic relationship, right? But like I mentioned, there's new evidence that supports that it may have been started off at least as a parasitic relationship. And so I need you to read this article by Wang and Wu from 2014. This is a perusal assignment, so you'll be annotating different parts of it. There's a lot of very specific cellular and chemical cellular respiration material in there that I don't need you to know the nitty-gritties of, so just focus on the topic of endosymbiosis theory and what that means for how mitochondria might have evolved. Okay. So the next thing I want to talk about is secondary endosymbiosis. And secondary endosymbiosis only happened with the chloroplast, right? But it evolved, right, or at least happened 3 times since, right, the chloroplast evolved. And I'll talk a little bit about the evidence for that at the end. Right, so secondary endosymbiosis is very similar to how primary endosymbiosis happened, except instead of it being a bacteria that evolved or that got engulfed by the eukaryote, now it is a small photosynthetic eukaryote that gets engulfed by a heterotrophic larger eukaryote. And I'm going to draw this out again on the next slide with you so we can talk about where all of those. All right, so we're going to go through it again, and let's start off with green. So green is going to be our original membrane that was the bacteria membrane, okay? And then we're going to do our outer membrane of that chloroplast, right? And then we have our, right, our cellular membrane for that eukaryote. So this would be a photosynthetic eukaryote, right? And this organelle right here, right, is our chloroplast. Chloroplast, right? From our primary endosymbiosis. Okay, so now what's going to happen is we are going to have a larger eukaryote, okay, that is a heterotrophic eukaryote, okay, and that eukaryote is going to engulf this photosynthetic eukaryote. Okay, so what we're going to get here, I'm drawing it bigger, okay, is our smaller eukaryote is going to be engulfed here, right? So we have our nucleus, we have that membrane from the original engulfing, right? And then we have our green membrane from when it was a bacterium, okay? So we have a secondary, right, engulfing, right? So what we're going to end up with here is, again, for whatever reason, that organism, right, that heterotrophic eukaryote did not break down that photosynthetic eukaryote that it engulfed, okay? So now what we get is, that is our secondary endosymbiosis, right? So secondary engulfing membrane, right? So it's our secondary engulfing membrane, right? And then notice that there are 2 blue membranes around this photosynthetic eukaryote over here, right? The first one here, this one on the outside, is that eukaryotic membrane, right? So this is our eukaryote membrane, right? So that one is going to be in, this is also not drawn to size, by the way. Okay, so this is our eukaryotic membrane, right, from our first endosymbiosis, right? And then we have our other blue membrane, right? That was our primary engulfing membrane, right, from primary endosymbiosis. And then we have our green membrane, right, from our original bacteria. Right? So now this eukaryote that engulfed that photosynthetic eukaryote now has a chloroplast with it that has 4 membranes around it. Okay, and that's one way for us to really know if it was from primary endosymbiosis or secondary endosymbiosis. Okay, so now let's talk about the supporting evidence really quick. All right, so the plastids in some of these protists have 4 membranes around their chloroplast, and we just went through all 4 of those. Okay. Others have remnants of organelles that are inside of those plastids, so they would be nucleus-like structures that were from the original eukaryote that engulfed that that was engulfed by that heterotrophic eukaryote. Okay, and then one other really important piece of evidence is that there are species of paramecium that have remnants of red algae in them. And really quick, I want to show you a phylogenetic tree. Okay, so here's a phylogenetic tree. It's right to my side, right? And you can see that primary endosymbiosis happened in Plantae, right? So primary endosymbiosis happened right here, okay? And all of these plants, right, have primary endosymbiotic derived chloroplast, right? But secondary endosymbiosis happened, right, at least we think, we have evidence that it happened 3 times, okay? One of those times was a red algae, right? And that red algae, right, somehow got engulfed by the ancestors of alveolata and Straminopila, right? And so paramecium happens to be a ciliate, right? So the ciliate's right there. And we find red algae remnants inside of lots of, or at least this specific paramecium species. And so that essentially should at least inform a little bit about the fact that these red algae were what were engulfed by the ancestors of alveolata and Straminopila, right? And then green algae, at least there's 2 instances of green algae being microscopic green algae being engulfed by the lineage that gave rise to euglenids, right? And the lineage that gave rise to these chloranophytes, okay? So we think that secondary endosymbiosis happened 3 times throughout evolution, right? And that's all I have for you today. So I will see you next time, and don't forget to read the paper about mitochondria and the evolution of mitochondria. All right, I'll see you next time. Bye.